Hello. Okay. Let's give a couple more minutes for people to come in. While we wait, if there are some of you that have things they want to present besides Alex Meyer today, I just want to let people speak up.
I did some SLAP 2 QC analysis. So
Okay. David, you have something to share?
I did some AI magic and made some figures. That are
Okay.
kind of nice, actually. But yeah.
We have two. Anyone else have something they want to share? Sarah?
You have some data to share on behalf of Yuhang because it's midnight in Malaysia. He's going to sleep, but he did some quality control plots for the electrophysiology data.
Okay, thank you. There's already plenty to cover. Anyone else have something they want to share today besides Alex? Okay, why don't we get started? So good morning, good afternoon, everyone. As usual, this meeting will be recorded, including for the Malaysian friends. So if anything occurred during the meeting you want to crop out, we'll do that. Just reach out before the end of the day. So our content today is the agenda is here. So Carter will start to make a brief update on data collection tables that we pushed. And then last week we discussed a number of updates from the group. We didn't have enough time for Alex, and so today he's going to present some of his analysis of audibles and tuning. And then we'll do a round. It seems like we probably should start with Sarah and Ido, and then we'll go with David. That works. Please keep it just to the point, because the point of this meeting is to make sure that people sort of exchange and continue the work asynchronously over the week. And we can have this meeting again next week where we continue the discussion. Okay, Carter, do you want to share an update on your work? I think that's here.
Yep. Yeah. Are you gonna share your screen? Okay.
Yeah, I think you can see.
Okay, everybody. So, yeah. So basically, we updated both the interactive and the static views of the kind of the overview of all of our sessions here for predictive processing. So we're kind of opening up to you guys to see, if this is clear, if it's useful for you guys. A couple of things to note. Let's see. So we have these QC tags, which basically for a failed session in general can be presumed to be the reason why the session failed. But sometimes, like for instance, Z drift, which would be tag 3, it's not severe enough for the session to be considered a fail. So you can see there's 3 on many sessions that are passing as well. And so we haven't updated the caption yet, but we'll need to update the caption to explain more clearly what the criteria are for the failure and for applying the QC tags. But hopefully this is useful for you guys to dig into your data. Like if you notice a session is missing running, for instance, this table could be used to confirm if that's expected, which it is in about 5 sessions. We also have the interactive view. If you want to click on that, Jerome. So we already had the animals in session table, but we added this data access table in here. I've also synchronized all these tables with what's on the public spreadsheet. So as we update the public spreadsheet, or if we update it, then this will have to be manually updated again. The data access table just has basically a much more compressed view of what's already on the public spreadsheet. So those of you who have viewed that, this will be more familiar to you. So it's basically just the links here. The most important link is the Dandy link, but then for each of the intermediate assets, there's the session and the spike sorted, for instance, for EFIS. But if you were to click on Mesoscope, modality.
Oh, yeah. We should
Right,
change that because SLAP2 is also 2 photon.
Right, yeah. Yes, we should. So yeah, then these are the intermediate assets there. In general, they're not going to be as useful for you, but the Dandy links will be here too.
Any questions on this update?
I have a question regarding the SLAP2 upload. So it seems to me that the most recent SLAP2 session uploaded on Dandy is from the 18th of December. And I didn't check yet, but I guess it only contains 1 zebra block instead of 2. So I wanted to ask the question what the current state of the remaining SLAP2 sessions is.
I'm sorry, your question was about a December session?
Yeah, so I'm doing the analysis of the zebra stimulus, and I think we decided to add a second zebra block after our last meeting in December. So I guess only the sessions from 2026 contain the second zebra block.
That makes sense.
And I saw that the most recent SLAP2 session uploaded on Dandy is from December. So I just wanted to know what the status of the other SLAP2 sessions is.
Yeah, so by my count, there are 3 like non-pilot SLAP2 sessions that I have yet to upload, but everything else should be there. Among the glutamate sessions, I should say, we have many voltage sessions that aren't yet ready. So those ones I'm working on. There's specific internal technical reasons why they haven't been uploaded yet that I need to iron out with the people from CodeOcean. But yeah, we're working on those.
Okay.
Is there a disconnect between what you see, Marcel, and what Carter see, or?
Yeah, so what's on this table or this sheet here? I mean, do you see most of those?
Sorry?
In this figure we're showing here, do you see the sessions that we display here? Or the mice?
I have to cross-check. So
Okay.
So far I only use the session tables for that.
Okay. Yeah. I can send in chat the 3 that I expect to not be there that I have not uploaded yet.
I mean, it's totally fine. I just work with whatever is uploaded so far, and then I guess when it comes to the final figures, we redo the analysis with another session. That's fine. Yeah.
On SLAP2, I do want to mention that we are going to collect more data in the fall to bolster the number. This was agreed with Casper's team. Sarah?
Yeah, just a quick suggestion. Because this display, I'm not sure, is it interactive already, or is it just the other one? Because it would be super useful if one was hovering over a given session block to actually know the date, because then, for instance, Marcel would have a much easier time checking, right, what's mapping onto the data he can access.
You can certainly try that. Yeah, we just have these tables and this static view, but we could make this a little more dynamic and have this on the top. Yeah, that's certainly possible.
Yeah, it's not bad. Yeah. David had a question in chat, which was about the table being updated. I've updated this table recently to answer your question briefly. Jerome, would you click on the data access sheet?
Can you see it?
Yeah. Yeah. Now, so you can see, David, now this says 163. So I think that's in line roughly of the passing sessions we see on the public spreadsheet. So yeah.
On SLAP 2, our plan is to try to do 6 more mice currently.
Yeah.
It's going to take some time, but that's our current plan.
Yeah, another note about your question, Marcel, is all of our glutamate sessions were done prior to January. The last one I think was December 18th, and I think that one might be the one you're pointing out that's on Dandy. Our voltage after that session, we started doing our voltage sessions for SLAP 2, and none of those ones are up or processed yet. So those are going to be a decent measure of time before we get them up.
Yeah, there were glutamate sessions that were attempted in the winter, and they all have different issues. And so we decided not to share those assets, Marcel.
But Jerome, I'm talking
so
about the voltage sessions.
Okay.
I think there were also glutamate that were tried later on, Carter, and that were aborted at earlier on. The microscope failed, and so we decided not to share it because it was too much issue to handle.
Yeah, there were three later glutamate sessions that all failed.
Okay, it's a good reminder that there's the glutamate in the voltage imaging. And like just to be sure, the Google Sheets still gets updated, right?
Yep.
Yeah, it's the background for this figure, right?
Yeah, so these figures get manually updated by me from the spreadsheet, and then the spreadsheet I update, maybe once or twice a week.
Okay, great.
Okay. Any more question for Carter? Okay, maybe Alex, do you want to take it from there?
Happy to. Jerome, can I post the link in the chat? So if you want to pull up the repo, you could show it.
You gonna share your screen, or you want me to share? Or
it's probably easiest if you just click on it. My laptop heats up and becomes loud if I have too many windows open. It's an old one.
Okay.
Okay.
Where do you want me to
go? So you can scroll down a little bit. So this is the repo I want to talk about today. So the way it came about is that Anthropic developed Claude Science or Claude.Science, as a lot of you probably know. And so as part of maybe the PR push, they invited scientists a couple of weeks ago to submit proposals and then selected about 10% of those and gave those scientists virtually unlimited tokens to use Claude Science. So I threw him in our hat and said, so we have this data, and maybe as part of this hackathon, we can use Claude Science and analyze the data. So what you see here is a repo that was established over a week or so of Claude Science, pretty much running continuously, obviously with me injecting once in a while. For those of you who don't know, the difference between Claude Science and let's say Claude Code or Codex, what you'd be using is that There's a few inventions here. Claude Science has already a few other sub-agents that serve as quote-unquote reviewers. So everything that it does, there's some other agents that look over everything and critique it. So this way, hallucinations and other issues are often caught. The context, the MCP is a little bit more scientific-minded. It does what the other coding AIs like to do, which to use your local machine to run the Python code and compute everything rather than in the cloud. But you can force it, like I did here, to push as much on GitHub and then double check that the code also executes rather than just having everything just in your IDE. Okay, so long story short, what you can see here is the README file. It's pretty long, so I want to go over that rather quickly. What I'm doing, and you can see right here, is that some of this is quite formatted, and that's because I went manually in the README file, and then I edited it. In other words, I translated from Claudish into English. So all of us know that if you have Claude or these AIs write a README, they use a lot of AI phrasing, like this was load bearing. And so I try to cut all of this out and rephrase it. So if you scroll down a little bit, Jerome, the first figure that you can see right here is like a summary figure. So because what I did is I told the AI that the overall goal is of our project that we'll look at what is in common across these different types of oddballs. Because the big motivation, obviously there's a lot we can do with the data, but the big motivation that was advertised is that we see is there one computation underlying all prediction in the cortex. Because that is obviously what makes predictive coding interesting. If there's 1,000 different ways of how the brain does predictive coding, then it's questionable if that's one thing at all. But the promise of predictive coding is like, here's this one algorithm, there's a prediction and error and a prediction error, and it happens across cortex. That makes it more interesting, right? So what the AI tried to do then is look at these 4 different types of oddball stimuli that we have and see if there's something in common. And so what it found is that with the exception of the sensory motor oddball, and there's a big asterisk here, it seems that we find positive, increased, enhanced responses. And so when we say responses, what does it mean? We have 3 different techniques. So to the right, you see that at least when we look at the neuropixels, so the spiking data, it's only spiking, not LFP, and the mesoscope data, which is the calcium signal, it seems that there's an elevation for 3 out of the 4 oddballs, again, with the asterisk for the 4th one. So this, by the way, almost certainly has bugs. I'm still going over all of that, but it is encouraging, I thought, nonetheless. And obviously, it immediately, to our scientists, raises all kinds of secondary questions, right? But so this is interesting and encouraging. So if you scroll down a little bit, I should mention that, I did use Codex, for example, to look over the repo and harden the analysis. So it's a lot of code at this point, obviously, with these limited context windows, but it did find certain issues. So what you see right here, some of it is trying to formally harden all of this and, find certain bugs. So I see Marcel has a question.
In the previous figure, what was the difference between the column A and B?
You mean the left and right side of the figure?
Yes.
The left side is only the spiking data, and then to the right side is a comparison between spiking and calcium imaging.
Okay. Mm-hmm.
So yeah, it's multidimensional, right? So we have different oddballs, but also different techniques. So it gets difficult to do it in 2 dimensions. So what the AI decided to do is let's only look at spiking. That's the easiest to analyze. That's the most data we have. And then for one of the oddballs, look across the 2 techniques. For SLAP2, we just don't have enough data yet. But so if you scroll down in the README, all of that gets explained a little bit. Okay, so so this is just a big overview. And I thought this is kind of encouraging. And also maybe a guide star for people that join the project. Like, what are we even trying to do? It's kind of why something like that in the end might be nice. Obviously, if we say, if we go this way, trying to find out if it's one computation, it's not enough to show that you get the same, signal increase. That doesn't mean it's one mechanism, right? So more work would be needed. But if there's one computation, that should also be a common signature. So I think that's encouraging. If you scroll down, the next thing was to look at subject by subject, so at the individual mice. I thought this is nice. So again, you see the 4 oddballs, and then each dot is a mouse. and you can see the variance across the subjects right there. And so you can see what's kind of interesting already with the sensory motor, that the reason that this seemed like a null result is that there's actually quite a spread. And I think that's where, people like us, not an AI, would get more interested in, like, maybe there's a difference between these two mouse populations and so on. So if you scroll a little bit further down, oh, I should mention that each of these figures you can see right here comes with a link to a notebook. So you can click the link, and then there's a notebook that you can run for free in the cloud in Colab, and it should spit out that figure. I see that Fasani has a question.
Hi, Alex. So I'm looking at the table that you were showing with the error type and prediction error index. And the one that especially caught my attention is the duration timing. Because I don't know if you're aware of the paper that we have, it's going to be published in a week, that we did not find in passive behavior, in passive visual behavior, we did not find any interval timing prediction error signals. So I'm very curious, like this 0.32. So do you, I want to basically know more about that.
Yeah, so as we scroll down, there will be more. So we can look at the actual time series. So this is just some summary. Yeah, we'll look at that.
Sounds good.
Okay, so if you scroll down a little bit. So one thing that's important is, again, what are you measuring there? And so this is obviously an index because of this whole problem of how do you know it's an oddball? How do you factor that into a control condition? So a lot of this is outlined here. But I think the time series are actually more interesting to most of us since they're easier to interpret. So if you keep scrolling, Jerome. So a lot of this here is still just AI producer with cleanup. Here is a summary table of the data as it was, I think, 2 weeks ago, that goes into this repo. So there might even be carded, there might even be more posted at this point, especially maybe for the SLAP. And so if so, I will go and update it accordingly. But you can see that there were only 2 sessions that I could use from the SLAP 2 data. There's 12 or so that we initially had that we all worked on to analyze the SLAP 2, but it didn't have the exact same paradigm. So there's only 2, and so that's why it doesn't show up in a lot of the data. And then some of the sessions you can see right here, there's 15 for the neuropixels and 21 for the mesoscope. Some of them came online as I was analyzing, and I was updating the data as we go. So this might be worth comparing to a similar table that Jerome put in the manuscript as we have it so far, in terms of when we publish the data, which NWB files or AWS files are we talking about? This is all strictly on the NWB files, by the way, on Dandy. I didn't touch the AWS. So if you keep scrolling, Jerome. So the reason that I had the AI write out gotchas and so on is that I thought that a lot of this README might be useful for anybody else who wants to use LMMs for their code, because you can basically just tell it, read that README file, and then use it as context to analyze the data, right? So if there's a session, for example, where there might be an outlier, then you don't need your LMM to rediscover that. So my idea was that if we have basically a skill file, this is like a shared skill file, that your element could read and immediately understand where the data is located, how to read it, and what individual sessions might be. So we'll get to the duration in a moment. So the first thing I wanted to do, what you see right here, is to replicate what we already had to see, catch any bugs. My thinking here is that if I'm a PI and I have a big lab, how do I know that my students didn't put a bug in the code? I can't read every single line of their code. I'd rather write it myself. So what I do as a PI is I tell them the same task 5 times. So I tell them, okay, look at the data this way, this way, this way, this way. And so if they each time write different code and they get the exact same result, then I know there's some coherence here. If they look at the same data in 2 different ways and they get very different results, it's clear that one of the pieces of code had a bug. So my idea here is, okay, let's start to do the receptive fields again, which I looked at before, other people here have looked at before, and they should look the exact same, pretty much, right? So depending on what your lookup table is and so on. So this is what I did right here. So these are the 3 techniques: neuropixels, SLAP, and mesoscope. and obviously pretty nice selectively picked receptor fields. What I haven't done here yet, which we discussed last time, or we might have an extra meeting on that, is to make sure that we can associate, let's say, neuropixels, each receptor field to a specific neuron in a specific animal at a specific session at a specific area and so on, so that we make sure that, okay, so for that exact neuron, Marcel finds the same receptor field as I find here, and then using a different AI, we find a different code. If we all get the exact same receptor field, that should boost confidence that none of us has a major bug in the code. So this was the idea right here, to look at receptor fields. Also, I thought this might be obviously a nice figure for our data paper, right? So because we say we have 3 techniques, and this is a way to compare the 3 techniques with the same receptor field analysis. So then next, if you scroll down, that's the orientation or direction tuning. Same idea. So if you keep scrolling a little bit further down, you get the polar plots. And so for each technique, looking at direction tuning, I say direction tuning rather than orientation tuning because we use drifting gratings. And so obviously in orientation tuning, you only get 180 degrees, but with the direction, you can get the 360. And that is what you see here. So then we can scroll further down. There's some summary statistics. All of that, again, please take with a grain of salt, but it doesn't look, it's a sanity check. It looks kind of as you would expect. So if you scroll further down, so now we get into the oddballs. And so the first thing here is, so now it goes oddball type by oddball type. So this the simplest type of oddball, where it's just a different orientation. And so it is looking, I think, at the neuropixels data only. And so it's trying to come up with all kinds of different statistics. And I haven't gone over this very carefully. Some of that we might want to present differently, but obviously we see an effect. So if you keep going down a little bit, I think the time series are what's most interesting, rather than the summary statistics. So here you can see actually some time series where you see the spiking response, the PSTH, basically the spiking response as a function of time for the oddball, and then the standard stimulus or another control. And what you see here is that elevation that you would expect. And if you keep scrolling, there's 4 PSTHs here. So what I was curious here is that in the old days, this is how we did spiking data. You show one neuron, then you show one session or one animal, and then you show all animals, so all sessions. So the idea is that rather than just solving this all with some abstract statistics, if we say that, let's say there's an elevated response to an oddball, you should see this for individual neurons, you should see this for individual animals, and you should see it across the whole cohort. And so that's the idea here of, I think if you scroll up a little bit, Jerome, this one at the bottom here, that's what this shows. So this shows PSTHs across these different levels, individual neurons, individual sessions, individual subjects, and across all subjects. Okay, so then if you scroll down a little bit more, then it does the same thing for different layers, so upper, middle, lower layers for different areas, primary visual cortex, and neighboring areas. Obviously, each of that is interesting and worth more looking into, and each of that you should take with a grain of salt, since I haven't completely reviewed the code yet, but all of that looks very promising. So if you keep scrolling. So now it looked at omission. Here again is summary statistics, which is interesting, but maybe not as revealing as if you keep scrolling a little bit around the time series themselves, so PSTH, which you can see here. So there are different time series that what happens during omission. And I think the interesting one is what you see towards the bottom here, which is, again, neuropixels to the left, mesoscope in the middle, and then SLAP to the right. And the interesting thing is that there's a difference between a mesoscope and the spiking. And so this I've seen a couple of times now using different approaches to kind of look at that omission. Again, could be a bug, could be that, I have expertise in spiking analysis, not in 2 photons, so maybe it's because, some photo bleaching or normalization I should do that haven't been done on something. But, there's a difference here that might be worth going
Can
after.
Can you point at the difference you want us to look at, Alex?
Yeah, so you see, if the NeuroPixels data you see in red, the oddball is elevated, and then sorry, you get an elevated response in the omission. If I
in the omission, because there's standard, oddball, and control. Oddball is omission for you, or no?
Hold on, the font is so small.
Maybe hold on. Is it better?
Actually, sorry. Keep scrolling down one more. There's another time series that comes right here.
This
sorry.
one, yeah.
Yeah, this is the one. This makes more sense. So what you see in orange is the omission, and so what you see in the spiking is that when there's nothing, there's an omission. You basically, we could argue now whether that's a response or not, but it's definitely less than if there's a stimulus coming on. And then in the mesoscope, you see that.
As we were discussing this, I just want to remind ourselves that the mesoscope was a pan-neuronal stain, and so you're seeing a mixture of exaggerated inhibitory cells. So NeuroPixels is a different bias, and so you're going to see many more pyramidals than you would with imaging. Which, yeah, inhibitory neurons have a different responses to oddball, is known. But yeah.
Fasani?
Yeah, okay. So I don't know how much we want to discuss science here, because the omission is something that I don't know how many of people here are familiar with our paper, but we digged into it with many different aspects. And we show that, yes, there is that response, but it has nothing to do with the temporal expectation signal. And the whole message of the paper is that not until you do all these different manipulations to show whether this is susceptible to temporal predictability or not, we can make the conclusion, yes, there is a response, but it's not necessarily an oddball response. It could be due to something else. So yeah, we can come back to this topic, but I'm saying that to make the conclusion that these are oddball responses, that would be something that I would say no, at least for this, until we see the careful analysis done.
I encourage you, Fazani, to engage with the data and
certainly,
test it
but
with
I'm
your
saying that
neurons.
the whole point of our paper was that when we present an oddball and we see a response, we should not jump into the conclusion that this is an oddball response. And I think that's what we're doing here, and I really want to be careful with that. But I would certainly engage more with the thing. I've been traveling in the past few weeks, but yeah. But I mean, the type of response we're seeing here is exactly what we saw. And now I know this is something else and we can talk about it. This is not an oddball response. For the omissions, the other stimulus, like oddball types, other people can chime in, but the omission is something that we are really looking into. Yeah.
Yeah, actually, you make me feel great, because I was more concerned that this could be something trivial. So I know you have this paper with the ramp, and so it looks like it. But so if you look at this and you say, yeah, that's what I would have expected, that's great, because again, I didn't code that from scratch. So
and then I don't think
news.
so. We actually saw it in both excitatory and inhibitory neurons. We saw it even in inhibitory, like not because yeah. So the NeuroPixels data also is not too different from what Alan had with the like is one of the supplementary figures there. So the ramp is there too, but it's just less. But I'm saying that none of these ramps are about oddball responses. So we have to talk about the data paper. So if we are trying to do some quick analysis and just presentation, we may be really risking saying something wrong. So
oh yeah. No, I'm not doing it. This is not meant to be
no,
published,
I understand
right?
that this is that, but also we have to, as a group, make a decision on how we like with the data, because a real scientific finding for each of these parts needs a lot of manipulation, more than just taking a response and saying that, oh, this increases relative to baseline, then there is an oddball response. And I don't think we have all of those manipulations here. So I guess this kind of guides us about how we want to do the oddball figure in the data paper. Maybe we just really leave it broad without making any concrete conclusions at the end that we found all false, because then we may be sending the wrong scientific message here.
I think that's totally fair. And so I'm actually more excited because, again, I totally agree with you. The actual scientific work only begins here, right? So you see in response, it looks different. It looks different in the SLAP. It looks different in the NeuroPixel. So now I feel like there's something to learn here. And so I'm excited because it already takes usually a graduate student or postdoc weeks to even get to this point.
Yeah.
And so it took me less than a week. So if this is all correct, this is really good news because we can actually now spend our time on what's much more interesting and much more important.
So this is all the cloud science. Did I get this right?
Yeah.
This is all right. But then you said that the code is not reviewed, but from what I'm hearing, you kind of trust it.
So it depends on what you call a code review, right? So I had an AI from a different company look over the code, and it found a whole bunch of things. And I repeat that process a couple of times until nothing pops up anymore. Obviously, the code executes. And then obviously, the other thing that we can do is look right here and see if this makes sense to us. Now, I would postulate that at least we had the level here of graduate student or postdoc level code, because human code will also have bugs. And so that's still possible here that there's some bugs. In fact, the bugs that are caught by, let's say, if I use a different AI and let this code be evaluated, they're often very minor bugs. It's nothing major, right? So it's often that, oh, here this variable is set to this value, and therefore maybe you took the median instead of the mean or something. That's the bugs, all right? But they're nothing that would really change the story by a lot. So that's why I have some confidence, but I still don't feel like this is new to me, probably to all of us, right? So I'm still very nervous. But the nice thing is that if you now want to work, for example, on, like you start with this, you have these different responses, how do they relate? You could basically just tell your AI to read this repo and take it from there. And the reason that this is valuable is that teaching the AI what this data is, what this project is. So, read our review paper, read the website that Jerome put together. You're easily talking $100 to $200 worth of AI work or tokens. It takes a lot of money for the AI to read all of that and learn all of that. Whereas this is compressed information that has been distilled out of that. So it's more effective if you feed this into an AI to keep going from there. And then again, my solution to how do you trust this code is, yes, I will manually review it, but I wouldn't even trust that because the bugs that the AI has found so far are bugs that easily a human reader will overlook. And so therefore I have this idea of, well, we should have these smoke tests, these sanity checks. Does this look at what we expect? We should have the convergence. And if Marcel writes code about receptor fields and the AI writes code for receptor fields, we should get the same receptor fields. And so we have this Bayesian approach. So we just basically let more and more credence to the code if the results make sense. And again, because even if a graduate student or postdoc would write the code or us, we can't fully rule out that it's completely bug-free, right?
Yeah.
That's how I sense it. Obviously, all of this is also open source. So, I mean, the code, everything is right there in the repo, which means that other people can also bug check the data. AI can bug check the data. And I think this is the best we can do. So that's my answer to that. I almost got hit by that hack for the Bitcoin. And so it was a minor mistake where a variable was set to true rather than false. That's set in the repo in GitHub for 5 years. And no human reviewer, despite people having millions of dollars on the line, ever caught that bug. It was an AI that caught the bug. So it made me rethink in terms of how much credence we lend to AI code versus human written code.
So if I want to add, so I've been thinking about all of this a lot too, with my new students using AI and all of that. And I totally agree with you. Our students also make lots of bugs, and I worry more and more that how do I know that I'm publishing a paper on a code that was not. And then you're right. The way we avoid it is exactly by, as a PI, I would ask multiple analysis that gets to the same question, and they have to be consistent. So if only one analysis is showing and not the other ones, that's where I know there is some inconsistency. And that could be either because of a bug in the code or because of the results that are not robust enough to show in multiple. And maybe that's exactly what we need to do here. So instead of the code review, we need to do how a PI reviews the students' analysis. And we say, look, no, go ahead and do that. test it with PCA, test it with this other one. Do we see the same thing? Yeah.
That's my thinking, yes. Okay, so let's scroll down a little bit so we can finish this up. So this is the omission. Great to hear that that looks reasonable. Okay, so this is where we get into the deviance data. So here, you, for example, you can see running versus rest. Like this is the locomotion state of the animal to look at the different. So basically, because we didn't have a clear finding for the sensory motor oddball, the AI started doing all of these analysis. Like what if the animal is running versus whether it's resting? So you could see the summary statistics to the left and then the time series to the right. And I think that's also reasonable, right? So we know that in the visual system of the mouse, is my understanding, there's more activity when the mouse is active. That's a blue compared to the orange here. Now if we scroll down. So this is where it started looking at the sensory motor oddball. And to be perfectly honest, this is so far away from what I'm familiar with that I have not even spent any time thinking about what we're looking there exactly. This may all be nonsense. If you scroll down a little bit further. So this is where it shows time series. Again, this is, I think, totally dominated by the locomotion state. So if you go further down. And then we get to the sequence mismatch, which is more interesting maybe. So basically all I'm saying is we can maybe completely bracket the sensory motor oddball for now. And it would be nice to have somebody like Kareem or maybe Sarah who are more familiar with that to look at that and think about whether these analyses even make any sense or what we might expect right here. I think that takes further thinking that I have had time to do so. So again, here the PSTHs I thought are quite interesting. I again here asked to get the different levels of looking at responses, and you can see that it's already kind of struggling to pull things out of the data since here the baseline periods are not aligned. And so for now, I just let all of this go. I feel like each of these would basically mean, okay, go in and take a look at why is that, and for example, should these be renormalized? So I just let all of this go for now. And then we come to the timing mismatch, which I think Fasani might be interested in. So here again is to the left, I think the more interesting comparison, the actual PSTH or time series. So that's comparing the omission to, so that's spiking, obviously. That's, yeah, I think it's basically the same as before. I forget which data it pulls over. But you can see, yes, basically when there's an omission, there's hardly a response compared to if you actually have a stimulus in blue. But there is something ramping up in red. And so, yes, that's consistent with what we had before. So if we could scroll down. It looked into the jitter control. So that I just, again, I haven't spent a whole lot of time thinking about all of these things because, again, that's far out of my wheelhouse. I do very simple visual stimulation. But I just want to mention that this is kind of interesting. That is something that Claude Science popped up. So it did these analysis and it has these reviewers, these other agents that are running in the background. And when it finishes the work and says, okay, I pushed this on the repo, then these other reviewers, they pop up. And so one of the reviewers, it's another AI, basically said, hey, there's actually a jitter control in there you should look at. So it wasn't me that said that, oh, we have this control condition. The AI caught it itself and introduced it. Roberto had... yeah, exactly. Okay, sensory motor mismatch is motor halt, motor emission, and motor orientation changes. Yeah, so as I said, I haven't even spent any time thinking about it. So you might find this interesting to read me, and again, take it with a grain of salt, but it might be the first time that somebody looked into that, and you might find this interesting. Now, if we scroll a little bit further down, again, 4 different PSTHs here, single level, subject level, all the way to grand average. Okay, if we scroll further down. And you can see it right here where it says reproduce, you can see these are individual Python notebooks that you can click at and run them either on your machine or on Google Colab. And They've been tested multiple times. They should execute. They should produce the exact same figures. So that's where you could do the code review. It's not one giant toolbox that would have to be reviewed. Each of these Python notebooks lives by itself, so it should be more easy to do. So here's, yeah, the different PSDHs for the feature oddball, the sequence, and then the duration. Let's go further down. Now, with each of these time series, me as a PI, right? So I'm not satisfied with that. I'd be like, okay, so what's your inclusion criteria? What is about mean versus median? What does percent change look like? What does the C-score look like? And so on. So as I said, this is just like a first pass. But obviously, I'm not saying this is anything publishable yet. This is just to like, okay, what's in the data? Can we get a quick overview and go beyond just the receptor feeds orientation tuning? So here I tried to do, again, laminar analyses. It used the CCF for that, and I would, like to use current source density to, for example, find out what's above or below layer 4, at least in the visual system, and I haven't gotten into that. There's some parts of the code that accommodate for LFP, but mostly this is literally just single unit spiking or the calcium imaging. So we can scroll further down. I think we might almost be done. Yeah, so this is trying to look at area by layer, because one of the predictions in the review that we had is that we might find that, certain layers are more involved across different areas. So it's trying to do that. But obviously, each time you abstract from the data and then abstract from that and then abstract from that, each time there's Gaussian error progression. So again, grain of salt, but I just wanted to give this a quick try to see if there's something that pops out. Okay, we can scroll down. Again, here, laminar differences again, looking at the different oddball types. The easiest, again, might be to interpret here the feature oddball for now. But again, like what was the inclusion criteria? Is this mean versus median, raw spiking counts versus indices, and so on? All of this needs further work. Then, here's a first look at the SLAP 2. This is just 2 paradigms. And so you can see basically each of these purple dots is one of the 2 sessions. And you can see, obviously, there's huge variance. The open circles is what we found in the, I think, the NeuroPixels data. So as a comparison, obviously, with 2 sessions, there's not much to make out of it. So this is also a good example that it's questionable whether in SLAP 2 we can just average all of the ROIs, right? So wouldn't we want to rather be like, okay, I want these synapses that are responsive or they have the receptor feed right there. None of this is done here, right? So this is the problem of like this one pass analysis. I would suspect if we limit our SLAP 2 analysis to interesting ROI, something else will pop out entirely here. Again, this is time series here from SLAP 2, but I think it suffers the same problem that these are just indiscriminate grand averages. Yeah, and then the AI got hung up here on I thought it was not too much of a difference between the mesoscope data and the NeuroPixels data. So you can see this, and this was more pronounced in primary visual cortex here, VisP at the top, and that's in VisI, where the data matched more. And so the AI started looking into, well, what could cause this difference between the spiking and the mesoscope data? To me, it's obviously because, again, we're just throwing everything in a bucket here. And so, any sensible analysis would be looking at the receptor feeds, the orientation, tuning, the responsiveness, and so on. Maybe, types of neurons. So I think this is coming about because we're quite indiscriminate here with the data. Not even sure in terms of maybe there's some motion or motor artifacts in there. So this would have to clean up. I'm not concerned that there's a difference. I'm not convinced that it is scientifically meaningful, but I let it do that thing. So we can scroll further down. Yeah, again, I don't like to look at statistics. I wanted to look at the time series, and if you look at the time series, the PSA is here, right here, you see there's bigger issues at heart. If the baseline doesn't even align, then you find different counts in the effect, the time window that we use for effect. So this needs more data, more analysis. Sorry. Okay. Here's another analysis trying to get that. I think, as I will go over this README, I will probably cut all of this out since I don't lend a lot of credence to it. We can scroll further down. Yeah, so it's looking here at maybe it's tuning and so on. It found something. I think none of this is really interesting. Yeah, so this is looking at annotation control. So this is maybe more interesting that it looks at the different control conditions that we have. You can scroll further down. We're almost at the end. This one I thought would be nice, maybe something like that. It wouldn't maybe necessarily have to be for the oddball, but I thought this is a kind of nice tree diagram of if we find an effect, rather than saying it's a significant effect across all animals, that you say, well, it's, you know, in that many neurons in this area, or those areas, and these animals, and so on, to kind of give a little bit of more an overview, you know, rather than lumping it all together. If we find something, again, take it with a grain of salt. But in the end, to kind of parcel the statistics more than just give, you know, a p-value. I thought this is kind of nice. Okay. Yeah, and I think that's about it. So at the very bottom of that README, it has a little bit more again about the code itself. Some of this is again maybe for another LMM to read, to take things from there, and that's it. So. Okay. Too long didn't read, so I wouldn't make too much of this, but the experiment that I ran here is, can I use an AI and get a first pass analysis in just a week, as maybe, you know, an undergrad or graduate student would do if I just put them on the data. And it seems that the receptive fields seem like they are reproducing, the orientation tuning is reproducing, the feature oddball response seems to be reproducing, and then the omission, I take Fasani's feedback as encouraging that might be reproducing. And so that's encouraging, and that we could maybe take it from there, with all the caveats that we discussed, that this is not a publishable finding on predictive coding yet.
Yeah, it seems like sort of has a lot of rough edges, but it sort of screened the land very quickly, and we will need to sort of lay out the individual follow-up that needs to be done very carefully. Yeah. It's 9:52. I want to keep a track of time. So what would be the best here? How do you think we should use this resource, Alex?
Well, I mean, first of all, I think what we discussed last time, that we want to slow all the way down and take it step by step. So those of us who do the receptor field analysis, let's make sure we converge. So we discussed that we get an ID for every receptor field, so we know which neuron it comes from. And then we see that even though we all wrote independent code, we get pretty much the same receptor field. So that should be a first step. And then we can do the same for orientation tuning. And I envision something similar then for the oddball analyses. So, and again, I will clean up this README, and on my mind, if other people want to use AI-assisted coding, this could serve as for you as a get-go starting point, rather than teaching the AI for long periods of time and a lot of money what this data is, where it's at, how to read it, and so on. You can just give it this README and it should be able to understand. Nicholas, you have a question?
Yes. Can you hear me? Okay.
Yes.
So with respect to the mesoscope metadata, perhaps you should double check that the layer assignments are correct. I was looking for the mesoscope metadata. I couldn't find layer assignments. I don't know if the suffix of the imaging plane is what refers to is what we should interpret as the layer assignment for the mesoscope data. For the neuropixels data, the layer assignments are included. Like, for instance, layer 2, 3, layer 5, layer 4, etc.
Yeah, mesoscope, we use a depth, so we know the depth from the surface, Nicholas, but layering is sometimes difficult to assign really precisely in imaging, because, you know, layers are continuous a little bit. This is based on our historical data, so we know the match between depth and certain layers. I expect in some cases that will be imprecise, but we can add documentation in the data release paper that explain how this could be done. Yeah.
I assume that with respect to the AI agents work, did you specify which imaging depths that correspond to which layers when you were comparing cortical layer depth with respect to the mesoscope and neuropixels data?
So I think most, if not all, the layer analyses should have been in neuropixels only. But again, I have not been that deeply into it. But yeah, let me double check that. But I thought this is coming from the neuropixels, and even there, as I said, I don't fully trust it because these layer assignments, Jerome, correct me if I'm wrong, but they're coming from an average brain.
That's right. So it's a
So
template.
in other words, it's an immaterial, non-existent platonic brain, right? And so what I'd rather be interested in is these individual brains, and that can be done with the Kern's density analysis. So at least in the early areas. And so, that's another interesting analysis. How much if we look at the initial Kern's density, the initial sink in the Kern's density, how much does that align the prediction from the average atlas?
So I will say that the analysis I showed last week concerning the similarity of what I call representational geometry across subjects and across recording sessions, I repeated that analysis for the NeuroPixels data set. I posted that to the discussion forum. I'll link that somewhere, but I ordered the x-axis using the cortical layer depth rather than the imaging plane depth. So I suppose if it's a scientific question, to what extent these different layers really differ from each other, and close to what extent the layers are being picked up by neuropixels or the depths being picked up by neuropixels are analogous to the different layers discriminated in the mesoscope data. I don't know. I suppose like the question I'm asking is if we're going to include a stability analysis that compares across recording sessions and subjects with respect to these different imaging depths, should we rely on the imaging depth or should we rely on the cortical layer assignment in the metadata?
I think a dedicated analysis should be done to validate the depth and match between modality, because I don't think you can assume the metadata is going to give you exactly what you want. So the kind of things you're suggesting make sense to me. Comparing the response across layers and depth in both modalities and seeing how they compare across, because NeuroPixel is a probe. It has many units along the shaft, and then the mesoscope is planes, and so it's a very different modality. So I think we'll have to sort of be careful there. I wouldn't trust them.
Yeah. Also, yeah, I would add one thing to that. Somebody who's worked on this whole idea of a cortical microcircuit and columns for a long time. The ground truth that you're aiming for probably doesn't even exist. So when we say there's 6 layers, it's not that every anatomist has ever agreed on that. And to exactly define the laminar boundaries in anatomy is a little bit of an art itself. Again, different anatomists wouldn't necessarily agree. And then there's also the fact that the mouse column is not quite as orderly as, let's say, the primate column. So so then it's like, should I use the imaging depth or should I use the assignment? My answer would probably be both, and see what you find. But I think that we should be careful when we say there's 6 layers, because it's biology. So for example, if you say, where does layer 6 end and the white matter start? Well, if you look in Nissl stains, there is no end of layer 6 and no start of the white layer, right? You just find less and less neurons, but they keep going all the way into the white layer, white matter, you find neurons. So there's when we have these diagrams in mind from papers, here's layer 1, layer 2, and so on, often when we look in the brain itself, these are not as clear cut.
Alex, sorry to cut you. It's then we haven't covered a number of people that had material, and I wonder if we should. I would like to ask people to think what we think about this. But what about everybody's that has things they want to share? Because this analysis is just going to explode in many directions, and with AI, we can do many more things. And so I might argue that it might be easier for us to coordinate if people submit 2 slides before the meeting and we go over them then because it's so much material. And so I would maybe we should do that next week, is just have the people sort of share 2 slides the day before the meeting that they want to sort of highlight, and then we review them at the beginning of the meeting so that we make sure that everyone has an opportunity to speak. Does that make sense? I think just 2 slides will be plenty. And then the second thing I might point out is it will be particularly helpful if the slides you generate are helpful for the data release paper. So we have a number of figures. There's a figure on unit identification. There's a figure on oddball analysis, and a figure on receptor fields that we need to finish. So to enable those follow-up studies that Alex started to sort of hint at. And so it would be great if next week any of you that have figures that could go in there, just submit your material the day before, and then we'll make sure to review this. Your work, Alex, it would be great if you could consider the 2 figures you might use for this oddball stimuli, because I think there's some of this work you did probably deserve to be in the manuscript in some shape, and probably the standard oddball in some fashion, but could be there. But that's what would be my preference, is that next week we ask everyone to bring in 2 figures that speak to this data release, and so that we can package the paper in the first shape, and then we might need some kind of analysis coordination meeting later on, because it's just going to go in so many directions. Do people agree with this plan? Does that sound productive? And you can share your 2 slides ahead of time in the forum so that we can have a discussion. Sarah?
You have just a quick question about one of the figures for the data release paper. So when you have an individual panel label there, do you think of a single individual plot, or do you think of multiple plots? Because I'm struggling, for instance, with quality metrics for electrophysiology. I can't have a single panel that captures the different metrics. And so I was trying to understand, for instance, if it's a 7E, is 7E a single plot, or is 7E a series of plots?
I think there can be a multiple plots, yeah.
Okay, because that's why we were not really sure what was meant to be visualized there. Yeah, okay.
But yeah, please try to submit 2 slides.
Yeah, sure.
For each panel, and I'm going to do the work of sort of integrating everyone's figure and ask questions, but we need to have that. Nikos?
Oh, yes. I would say, like, we're also meeting, we have a meeting scheduled for this Friday to converge on the receptive field analysis, to see how all of our analyses complement each other and can fit into the, or would fit into the data release paper.
Yeah, that's great. Can you share it so that people who wants to join can on the forum or somewhere? Did you share it? Or
Yes,
not sure?
yes. There's a discussion thread where we have a poll, and we are scheduling the meeting for this Friday at
Great.
3 p.m. CEST, Central Time.
Yeah. Okay.
I suppose I can share it in the chat, the discussion.
Is there any other question for next week? We're probably going to have too much material moving forward for any meeting, so it's a good thing.
Jerome, just be quick, for the figures that you want to put in here, do you want them, do you need code? Because the other figures are interactive, right? They're pulling code. Do you need code or just a vector graphic? What do you envision what people contribute?
Ideally, it's code that regenerate the figure that I can integrate into the manuscript. Ideally, it works from S3 files or NWB files on Dandy. So I should be able to run it. Any other comments or questions? And please share any things you've done on the forum. I'll do my best to interact, and I invite you all to continue the discussions. We're all sort of learning how AI can help us, and it's a new world. Any questions, feedback? I think we should be good to depart, and then we'll meet again next week, going through those 2 slides. Thank you.
Bye.
Thank you. Bye-bye.
Bye.